Okay, we're going to get started. Good afternoon, everyone, and welcome to the third annual UCLA Children's Discovery and Innovation Institute Symposium. It's titled Generating Cross-Campus Collaborations to Advance Translational Child Health Research. I think we have an amazing afternoon planned. However, before I begin with our keynote speaker, I want to acknowledge key individuals who made this happen and have worked on our behalf the whole year and beyond. And that is under the leadership of Drs. Peter Szilagyi and Martin Martin. We have the CDI Steering Committee of Barbara Moshiski, Paul Chung, Isidro Soluski, Bridget Gompertz, Candice Wilkinson, and Miss Veronica Germer for their organization of this exceptional afternoon. So I want to begin by giving them a very big hand, really. It's a yeoman's amount of work. But it certainly promises to be a great afternoon because in addition to our keynote speaker, Dr. Atul Butte, we have two other outstanding invited speakers from UCLA, Drs. Carol Mangione and Susan Buchheimer, and our NICHD-sponsored K-12 Child Health Research Center scholars will present their work, followed by posters demonstrating the research that fellows and faculty who've undertaken are going to be there. So please stay till the end, give them feedback, and please interact. Hopefully it will be a rich scientific session. So now coming to our keynote speaker, it gives me great pleasure to introduce Dr. Atul Butte, who's professor of pediatrics and the inaugural director of the Institute of Computational Health Sciences at the University of California, San Francisco. So Dr. Butte is also the executive director of clinical informatics across the 6 UC medical schools and medical centers. He graduated in computer science at Brown and worked for a while at Apple and Microsoft. He then went on to receive a master's in medical science and medical degree from Brown. He then completed his training in pediatrics and pediatric endocrinology at the Boston Children's Hospital. He also then earned a master's in medical informatics from MIT and a PhD in health science and technology from Harvard and MIT. His research focus has been on translational bioinformatics and the development of analytic, storage, and interpretive methods to optimize the transformation of increasingly voluminous genomic, biological, and clinical data into diagnostics and therapeutics for the clinician. So before he went to UCSF, he was at Stanford for a while. And what he says is there is an urgent need to take what we have learned in our genome era and use it to create a new system of precision medicine, delivering the best preventive or therapeutic interventions at the right time to the right patients. His lab builds and applies tools that convert trillions of points of molecular, clinical, and epidemiological data measured by researchers and clinicians over the past decade, and now commonly it's termed bed data, into diagnostics and therapeutics while providing new insights into diseases. Dr. Butte will highlight his lab's research work, including the use of publicly available molecular measurements to find new uses for drugs, such as new therapies for autoimmune diseases and cancer, discovering new druggable targets in disease, while also using clinical and genomic data that have resulted from clinical trials towards discovering new diagnostics, including blood tests for complications during pregnancy. He's authored nearly 200 plus publications, with research repeatedly featured in Wired magazine, the New York Times, and the Wall Street Journal. He was elected into the National Academy of Medicine and was recognized by the White House as an open science champion of change for promoting science through publicly available data. He's also the founder of 3 investor-backed data-driven companies, one of which is discovering diagnostics for pregnancy complications, and another finding new uses for drugs through open molecular data. He's also the principal investigator of the California Initiative to Advance Precision Medicine and the principal investigator for IMPORT, the Clinical and Molecular Data Repository for the National Institute of Allergy and Infectious Diseases. So please help me welcome our keynote speaker to Southern California and to the podium. The title of his talk is on the slide.
Thank you very much. That was more than generous for an introduction. Thanks for having me here. And of course, I'm a University of California employee, so you don't even have to clap at this point. So first of all, I'm a medical doctor, obviously presenting to a medical audience, so I always have to start with my conflicts of interest. I have just a few. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You might not want to believe another word I say over the next hour. I wouldn't blame you. But I'm most proud of the bottom right. Those are all the companies started by my students. More than half my graduate students now start companies, even if they go into academia. And they do it with the most amazing platform in the world, and that's data. It's often big data. It's often open big data. So I'm going to show you how they did it, and I'm going to show you how I do it, and maybe I'm going to convince you this is the most amazing time to be in biomedical research. Now, in my lab, we do have this saying that if you want to change the world, you can't just keep writing papers about it. You do eventually have to tell the world about your discoveries. If there's something you can file a patent on, you should file it. And if nobody wants to license that, so you're going to have to start the company yourself. So we practice that. You're going to see. To me, it is absolutely not taboo to talk about companies in an academic institute. So you're going to hear about companies throughout this talk. So for a lot of folks, this is their slide of shame. For me, this is my slide of significance. This is what I'm doing to actually make a difference for patients. All right, so if you haven't heard, we're in the middle of this data deluge. My favorite example of this is the cover of The Economist magazine. This is from about 4 or 5 years ago now. This guy's collecting ones and zeros with his umbrella here. And this is the issue where they announced that the human species is now generating 2 zettabytes of data every year. If you forget your metric prefixes, it goes kilo, mega, giga, terabyte, petabyte, exabyte, zettabyte. And then, of course, the following year, it's 4 zettabytes of data because this number is doubling every year or two. I'll be the first to admit most of these zettabytes of data are these YouTube videos of kittens playing with pianos and yarn, entertainment value, perhaps no scientific value, but there is scientific data in the zettabytes. So, for example, the Large Hadron Collider in Europe generated petabytes of data to help those scientists find new subatomic particles. But my favorite example, though, is NASA. So NASA announced about a year or two ago that they have so many telescopes taking pictures of the sky and they're sending more into space, right? That by the end of this decade, they're thinking they're probably going to generate an exabyte of data every day, just in pictures of the sky. Now, but NASA already admits, though, that even today, they take so many pictures of the sky that there are actually not enough astronomers on the whole planet to look at all these pictures, believe it or not. So what do you do when you have too much scientific data? Well, NASA, they actually get their friends on the internet to help. So that's called crowdsourcing, right? So you can go to Galaxy Zoo, take a test, and then you get to assign, is this a star, is this a galaxy, right? So you're the person who does it as a citizen scientist. So even NASA admits that when you have too much scientific data, you need a new generation, a new cohort of scientists, because there are not enough scientists out there to actually practice the science anymore. And of course, I'm going to tell you we're there in biomedicine as well. But we have so much data now that actually this was a very influential paper to me from Chris Anderson, 2008. It was in Wired magazine, not the most scientific of journals. But Chris Anderson wrote in 2008 that we have so much scientific data now that science itself is getting obsolete. Now, how do you say science is getting obsolete? Well, it's actually a scientific method, okay? You remember, in fact, we're all taught in 3rd or 4th grade what the scientific method is. You come up with a question or a hypothesis, and then you go make measurements to answer that question, right? To address the hypothesis. Question, and then go make measurements. But we're now in this world where we already have the measurements. We already have the data. The new magic is figuring out what's the question I want to ask. What's the killer question here that everyone's been dying to know the answer to, and no one realizes we can actually ask and answer that question with this data already. That is the hard part now. Ask people in my lab, 99% of the work in my lab is figuring out what's the question. It's not writing code. It's not getting the databases and data munging. It's what do you want to do with all this data? That is still the hard part here. The random computer scientists do not know what to do with all this data. right? But the computationally empowered biomedical scientist does know what to do because they know what the unmet need is still. So I actually got a lot of influence from this early paper. And of course, we're in this data delusion biomedicine too. Now, I could have shown you any kind of cool nifty gadget up here. I'm showing you instead a gene expression microarray from Affymetrix. Now, I could have shown a DNA sequencer, mass spec, proteomics. I could have showed you, any of those kind of cool technologies. I show you this because this is how I got my start about 17 years ago. Scarily, it's been 17 years since I started my fellowship, I guess. And I was lucky enough to enter a lab where these had just gotten invented. We were analyzing the first, 60 of these different cancer cell lines. And those are amazing. You put any sample on them, and you get a readout of the entire genome. All the different genes and how much is expressed. In fact, they're so cheap now, I can carry one in every suit. Okay, so this is what they actually look like here. Big data comes from small packages, just like this. And it's astounding. I mean, they're only a couple $100. In fact, everyone runs these. In fact, if you get tired of measuring them one by one, we actually measure them in the 96 well plate format. And that's so cheap, I can carry that in my bag. Okay, in fact, this one's kind of falling apart. I need to get a new one. In fact, when you get tired of measuring them 96 at a time, they now come in a 384 well format. And that's so cheap, I have that in my bag. I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates. And we don't even use these plates anymore because we moved on to RNA-seq. In fact, Affymetrix doesn't even exist anymore. They just got acquired, I think, a couple months ago by Thermo Fisher or some random company. They'll still keep making these chips. What an amazing time. Everyone runs these types of measurements. Again, I could have shown you DNA sequencers. I could have shown you anything. But this is how fast exponentially growing this kind of data is. And what happened is when I got my start in 1998, 1999, that's when I started my fellowship, started doing some research. Everyone started to get these chips. Everyone runs them. It's not a big deal. And everyone started to write a paper. Here's a list of genes that are changing in this disease and that disease. Here's cancer. Here's heart disease. Here's a list of genes. There's a list of genes. They all started to submit their papers to the journals. And what did the journals do? They got overwhelmed. And they said, hold on a second, we're getting too many of these papers. No more papers with these gene chips in it. No more papers. Unless you deposit this data into an international repository where the reviewers can double check the math. It's probably important to do that. We're getting closer to the clinic. And maybe others can use these samples for something else. Okay, that was the idea from the top tier journals. Now, the precedent for this goes back to something called GenBank. Any of you heard of GenBank? It's a next door neighbor to PubMed. There are a couple of hands up there. Everyone's used PubMed in this audience. Next door to that is GenBank. It's one of the tabs up there. GenBank, I think, is 38 years old now, right? The very first papers with the DNA sequence, nobody wants to type in AGTC again, right? You're sending tapes to each other, even way before disks, certainly before the World Wide Web. And so the precedent is set as if enough scientists use a molecular technology, eventually you got to share all that data with everyone else. Fast forward to August of 2012. This article came out in Nature, featured my lab and a couple others. We hit a milestone of a million of these samples publicly available. You don't even need a user ID and password. 1 million samples. Up from 0 when I started my fellowship and research, up to 1 million. Now, if you have really sharp eyes, you can notice, boy, it's kind of sad. It looks like it's slowing down there at the end, right? You see the curve there? And then you realize they only counted half the year. This was August of 2012. Still doubling every 4 years. In fact, we're just a couple thousand shy of 2 million samples publicly available. 80-90% of these are human samples, some cancer, some biopsy, some something. Some mouse model, rat model, someone ran an experiment, someone digitized it, got all the readouts, and deposited all that data. So what does that mean? Well, you can get lost in zettabytes and millions and billions, so let me make this really concrete, right? Any of you who want to do science today, who know about molecules and diseases, you can go to these websites and type in whatever disease you want. Pulmonary hypertension or medulloblastoma or colon cancer, whatever disease you want. You go to these websites. You start with the data instead of a pipetter because the data's already been run by someone here, right? Oh, forget about you guys. Even any high school kid that needs to do a science fair project, even she can go to these websites. Let's say she wants to do a science fair project on breast cancer. She can go to these websites. So this is the European one. This is the NIH one. Doesn't matter which one you go to. Type in breast cancer, hit search, find and download 60,000 samples of breast cancer. About as easily as she can find a song on iTunes today. 59,000, yeah, 60. This is already a couple months old. I think it's like close to 70,000 samples of just breast cancer freely available. Now, a couple things about 60,000 samples of breast cancer. So what's so magical about having 60,000 samples of breast cancer? If you haven't figured it out yet, that's more samples of breast cancer than any breast cancer researcher will ever have in their lab. Because every one of them eventually has to submit data into the middle. And whoever figures out that middle database now has more data than any one researcher in the world, right? Because they all eventually have to share that data. And maybe it's not breast cancer, it's colon cancer, it's medulloblastoma, it's whatever disease you want. Probably someone has already studied it with these chips already. And all that data is just sitting there waiting for you. Because what do people do when they get a grant to get more data? They get another grant to get more data, right? Very few people actually go to these repositories to download the data and actually use it. Now, the number I actually really like is 2445. Let's call that 2,400. 2,400 labs and research groups have contributed data here. Let me tell you what a ridiculous number that is, okay? Let's pretend I'm a brand new researcher in breast cancer. I know nothing, I'm a newbie. And I'm writing an NIH grant to study breast cancer. And you're all in my study section. And I'm gonna write in my grant, I propose to you, that I'm gonna get 2,400 of the best breast cancer labs in the world to get their best characterized patients, to run them on the latest measurement technologies, and I propose to you, I'm gonna get them all to give me that data for free. For free. If I actually wrote that down in an NIH grant, you would laugh me out of the room. Some of you are already smiling, I can see you, right? You laugh me out of the room. Yet here it is. 2,400 labs are there helping, and they're doing it for free. So when I think of public big data like that, the phrase I use is retroactive crowdsourcing. What does that mean? 2,400 labs are there to help you, and they don't even know they've been helping you. Actually, 2,400 labs are there to help you, and you don't even know they're there to help you. And if for some reason 60,000 samples of breast cancer is still not enough for you, just wait another couple of years, it'll be 100,000 samples. In this hour, we'll add another 150 samples, just on average. That's how many are just getting piled into these repositories. What an amazing time to be in science. Yeah, I know funding is going down and all of that, and then they give you 1 million samples to get started for free, right? If you choose to use that. Now, if you have any doubts a high school kid can do it, here's the high school kid that did it. Okay, this is Brittany Wenger from Sarasota, Florida. I've yet to meet her. I talk about her in all my talks. Here she is over here because she won the top Google Science Fair competition when she was 17 because she built an artificial brain to diagnose breast cancer. So she took immunohistochemistry data, immunohistopath, qPCR data, and built a neural network. Okay, that's what a brain means there, and was able to make a diagnostic out of that. I guess she was bored, so the following year she did this for leukemia. She was in the paper for that as well. And of course, what I mean is if a high school kid can do this, we all better learn how to do this now, right? You're not going to go to the computational person to get you started. We're all going to need to do this. Maybe you don't realize how much coding is taking over the world. You know, in the state of California, of course, we're ahead again. You know, they're getting rid of cursive in elementary school and replacing it with coding, okay? This is true, okay? You don't need those cursive licenses anymore in 3rd grade, right? Because we're going to start at age 7 and teach kids how to code, right? So that's who's coming behind you, okay? We all have to learn how to do some of this. I'm not saying to be a professional programmer, but you gotta wanna know what to do with data today, because even the high school kids are coming. So this is a new world today for us. So, you know, that's what high school kids are doing with this. And what are we going to do with this? We're heading towards precision medicine, right? So, of course, precision medicine, everyone's talking about precision medicine. I love that phrase. Even President Obama uses it now, of course. So everyone's talking about doing precision medicine. And it is interesting. I mean, to me, what's the definition? My definition of precision medicine is the customization of medical care, customization of care for a patient, given the measurements I'm getting from that patient. They could be molecular, could be behavioral, could be from your smartphone, right? But it's not just you, it's also the measurements I'm getting from the entire rest of the population. And my experience in taking care of the rest of the population. What did we do that worked? What didn't work? So I got all this data from everyone else and I got to apply it to you. And for that to happen, I need basic science to connect all those dots, to get the measurements, to get the connections between this kind of measurement, that's all basic science too. So it's a customization from you and everyone else. To me, that's precision medicine. Now, I'll be honest with you, precision medicine as a phrase kind of makes doctors nervous. It's true. Because by definition, if we're moving into this era of precision medicine, that means the last thing we were doing was not precise medicine, right? By definition, right? So this actually kind of gets doctors kind of weird. How dare you say we were not in precision medicine or personalized medicine? So actually I started to think about how do you actually measure the precision of a medicine, right? How do you say it's precise or not? Well, to me, one way to think about the precision of a medicine is just to think about how many diseases we have, or how do we classify all our diseases. Our patients, we put them into these kind of little buckets of, these little pigeonholes. You have that disease or this disease. And the field of that is called nosology. Nosology has nothing to do with your nose. It's a systematic classification of diseases. And so I think a lot about how we classify diseases. In fact, this idea goes way back to Linnaeus. This is who that's shown on the picture here. Linnaeus is that famous guy. Remember, you all learned it in high school or elementary school. He's the guy who put all the diseases and all the species into a taxonomical tree. Right? Remember, kingdom, phylum, class, order, family, genus, species. Do you remember that? You all had a mnemonic for that. Some were clean, some were dirty. Don't repeat them here. It was on a test for all of you at one point. You had to know this. That's what Linnaeus did. But Linnaeus was also the first guy to put diseases into a taxonomical tree as well. So he published a Genera Morborum. This is in the 1700s. He's not remembered for this because he got it completely wrong, okay? You can see these are his on a broad categories. You can see how they're trying to make a tree with the fonts here. That's the best they could do. And why did he get it wrong? Well, he said, put the fevers over here, fevers with urinary problems over here, something wrong, mental diseases over there. He just completely got the thing mixed up here. To give him some credit, though, this was only 80 years after Galileo was under house arrest for saying the sun was the center of the solar system, so it's not bad for 80 years. But he got the disease, he got the species so correct, so he's remembered for that. But what he launched is for 200 years. If you were a learned physician or learned philosopher, the thing to do was to make your own classification of diseases. And we still have these classification of diseases, right? Any of you heard of the ICD-9 codes and ICD-10 codes? Of course, you all have here, right? In fact, we just moved to ICD-10, right? So those ICD codes come from William Farr and the Royal College of Physicians in the 1860s, okay? That's the first of the batch of ICD codes. So to think about it, we're moving now to the 10th edition of something that came out after the American Civil War. That's how bad precision, and it has to do with money now. Did we get reimbursed the right amount? It has nothing to do with science. Let's fast forward a little bit to ICD-2. So ICD-2 was 100 years ago. Manual and International Classification of Causes of Death. You can get these off Google Books. In fact, I have a copy of this one here, which I just carry in my bag. This is what it actually looks like. Second revision, Paris, 1909. So it's about 100 years ago here. And when doctors die, this becomes their trash. You know how we save books. You can buy these off Amazon for pretty cheap. And it's amazing. When I read through this book, it's amazing how it's written like our current code books. It's 100 years ago, right? It literally says, code 39 is code cancer of the buccal cavity, which is cancer of the cheek, right? Code 40, cancer of stomach and liver, intestines, skin. I mean, it looks like our current code books. Use a number, here's the code, here's what it's for. And then 100 years ago, they still did with the codes back then what we still do with the codes today. We're running out of numbers, put all the other cancers in 45, all right? Not otherwise specified, right? Including literally lung cancer and brain cancer. I kid you not, it's in the book. They were not worth distinguishing from each other 100 years ago. In fact, you can learn a lot about medicine, about what you put into not otherwise specified. It's all the stuff that we cannot diagnose, cannot treat, not even worth distinguishing from each other, right? It's all the stuff we're kind of embarrassed about. I like studying what's in not otherwise specified here. But isn't lung cancer the number one cancer killer today? Was cancer not otherwise specified 100 years ago? And literally, though, this international standard goes on. I mean, I flagged the page here if you're really curious. This international standard has to say that if you happen to run into a patient that died because of a visitation from God, be sure to use code 189, okay? Now, the same way you're laughing at this, they're going to be laughing at us in 20 years, right? Oh, those silly fools didn't know this bowel disease and this joint disease were the same thing, right? They're going to be laughing at us because we classify diseases based on the kind of doctor that takes care of them today, which is completely irrelevant from the molecular side. So the visual to think about is replacing this with this, right? That's the kind of idea here. Now, of course, we're moving ahead. We've used ICD-9 for the last 30 years. And last year, we moved to ICD-10, finally. And actually, all the physicians seem like they're against ICD-10 because there's just too many codes, right? They're making fun of the codes here. There's even a code that if you're surfing and hit by a whale, there's a code for that. There were a lot of memes going around the internet making fun of ICD-10. This was my favorite. I don't always get sucked into a jet engine, but when I do, I use ICD-10 code V97.33XD. There's a code for getting sucked into a jet engine. Now, of course, I have a lot of friends in San Diego, especially at UCSD. So I've given this talk at UCSD a couple times. And actually, they've come up to me at the end and said, indeed, it is possible if you're working at a naval base on an aircraft carrier, you can actually get sucked into a jet engine here, right? But this is the chronic form of the disease. I can imagine getting sucked in once, but not twice, okay? But there's a code for that, right? There's a code for that, right? So people made fun of ICD-10, but what we should have been doing is we should have been making fun of ICD-9, okay? So let's just talk about lung cancer again, just showing you 100 years later. This is ICD-9's code for lung cancer, 162.9, all right? And these are all the diseases that fall in lung cancer. Do we take into account the latest in gene sequencing, EGFR sequencing to figure out what drug to use? Of course not. Do we take into account the latest molecular pathology to figure out subtypes of lung cancer? Of course not. Do we even take into account the microscope to distinguish large cell from squamous from adenocarcinoma? Of course not. Do you know how we used to classify lung cancer in ICD-9? Left lung or right lung? Upper, middle, lower lobe. I'm not kidding you. If you ever need a definition of imprecise medicine, here it is. And we have got to stop making fun of ICD-10. That's not even going to solve all of this. We have got to get to a mode as physicians that we want to accurately tell the world, especially each other, what do we think this patient has? And more importantly now, use the right codes to tell the computer what we think this patient has, because we're going to need the computer's help as we go forward to know which tests to order, how to interpret them, and all the rest. So let's stop poking fun at ICD-10. It doesn't even solve all of these problems yet. And that's where we got to get to for precision medicine. So I'm just going to give you a couple anecdotes of what we've been doing for precision medicine here, just a couple of diagnostic therapeutic things. So my first big NIH grant when I was at Stanford was go get every human disease studied by microarrays. Why pick diabetes? Why cancer? Just go get all of them, right? I mean, and when I moved to Stanford 11 years ago, we had a whopping 25,000 arrays publicly available. What am I going to do with all of those, right? Now we're at 2 million. Right? And so we started just collecting disease and healthy. We collected experiments where people looked at disease and healthy samples at the same time. And so we can, each box here might be a gene, and the colors represent how much of that gene. We can calculate these differences or signatures. People have been doing that for more than a decade. It's not a big deal. Marina helped me make the slide. She did a lot of the initial work. But you know what? I could pick any disease. I don't just have one experiment. I might have dozens, hundreds, thousands, right? I showed you thousands for breast cancer. And what we do is we can put them all together. And it's kind of a meta-analysis. So I don't trust any one of these experiments, but I trust what I'm going to get in common, right? What are they all seeing that's in common here? That's kind of thinking here. And what's the first thing we're going to do with all this data? Well, we wanted to create new diagnostics. So what's a diagnostic? It helps us tell if a patient has a disease. We have imaging diagnostics. I love protein diagnostics. I love proteins. I know for the aficionados in the audience for a second, here's the central dogma. DNA codes for RNA, codes for protein. Everyone kind of knows that. I know these chips really help you measure RNA because they're kind of easy to measure, but I love protein tests because I still remember my days in the primary care clinic that if I had a child with a fever and a sore throat, squirming all over the place, somehow I could stick this Q-tip back there, put some drops, and it could tell me if there's strep or not there, right? That's called an ELISA, an enzyme-linked immunosorbent assay. That kind of works in a desert. I mean, you don't even need a refrigerator or anything. I love those kind of tests. Compared to RNA tests, I love RNA. You can make companies with RNA tests, but if you just even look at RNA the wrong way, it starts to degrade on you. Okay? If you spent time in the lab, you know what I mean. Because our body gives off RNases, it kind of dissolves the RNA around us, because to our body, an RNA might be a virus. So I love protein tests. And so if we see a lot of the RNA changing, we're going to just chase down the protein. So we had a couple of these, we had a bunch of these. One was a cancer marker paper for medulloblastoma. The surgeons told us that, when they're operating on a kid with medulloblastoma, it's still sometimes hard to tell what's the cancer, what's the normal brain. And they want to keep as much normal brain as they can. So they said, well, can you come up with some kind of paint that we can just paint the operative field, turn off the lights and have it glow where the cancer is and not the normal brain? I'm like, how the hell do you design a paint like that? Well, you just go to the public repositories, download everything you can on medulloblastoma. So you got all the positive signal. Right? Then download glial cells, astrocytes, neural cells, and you subtract out what you see there, figure out what are the cell surface proteins, and then chase them down. And so here are two of them that we stained. You can see it's darker here than here. That's an example of a cancer marker here. We care about transplant rejection markers. Here, when a new organ goes into a kid or an adult, some days the patient wakes up and says, the body feels like this isn't part of me. I got to get rid of this thing. That's acute rejection. It's not painful. Patients don't know this is happening, so we have to stick long needles in to kind of make sure it's not happening, right, to kind of screen them. I wanted a blood test for transplant rejection, but you know what? I wanted a universal one. I don't care which organ went in, whether it was heart, kidney, liver. If it's rejecting, I want the same test to be upregulated. So we do a massive Venn diagram. What's in common with all these public experiments? You can see there's a paper several years old now. You can see here's a marker that's up in kidney, liver, and heart rejection compared to normal transplant. And it's in the serum as well. So we did a bunch of these. The one I'll spend a little bit of time talking about is preeclampsia. Now, everyone in this audience probably knows what preeclampsia is. This, to me, is an extremely annoying disease. Most Americans only know about preeclampsia because they watch Downton Abbey, and one of the characters dies of this. Spoiler alert if you haven't watched this, right? But that's usually why most Americans even have heard of something like this. Of course, it affects a lot of people. Still how husbands lose wives. It affected my wife. My daughter was born 12 weeks early, 1 pound. She's totally fine now, 1 3/4 pounds. She's totally fine. But this disease really annoys the hell out of me because it falls right between the cracks of medicine. I love my friends obstetricians, but you know what they say is the delivery is the cure. Just take the baby out. Some mag sulfate, the mother will be better in 48 hours or so. But then we pediatricians have to keep these babies alive for months, right? These tiny little things. And so it falls right between the cracks because we in pediatrics can't really tell them to keep the baby in. How can you do that? We can do our best. But when the baby's coming out, we got to do our best to solve this, or at least keep them alive. There are actually 4 drugs in trials right now for preeclampsia, but the diagnostic is basically something we've used for 50 plus years, right? Urine proteins, kind of non-specific in today's obese America, right? I mean, not the most specific thing. So we wanted a specific test for preeclampsia. Linda Liu was a grad student who started the work. Bruce did the protein work. Matt Cooper got involved later. And how do we start? Well, let's just go to the repositories, type in preterm birth or preeclampsia, 266 experiments waiting for you. I don't mean 266 samples. I mean 266 collections of samples, right? Here are 299 human samples, 249, 154, 143, 104. Someone's already studied this, some tissue, some muscle, some anything. All that data can be downloaded. And the basic methodology here is download all the good ones that you can and just intersect them. What are they all seeing in common? What's commonly upregulated? I don't trust any one of these experiments, but I trust what they get in common. It's called wisdom of the crowd. So you chase it down. We've got the RNA, chase down the protein. Here you go. Here's the protein. Here's one protein called hemopexin. It's up in preeclampsia compared to normal, healthy, pregnant women here. So March Dimes funded this work in the beginning, and Spark was a CTSI seed grant. They call it Spark at Stanford. You could just see, here's one of, I think, 6 or 7 that we found. A bunch of papers published there. And what do you do next in Silicon Valley? You got to start a company on this, right? And so you're going to see a couple of these arrows here, right? So let's just go through the arrow here. We start with the unmet need. Remember, we don't write a line of computer code unless we have that question, right? If we don't know what we're doing, we don't write a line of code. Public data is available. March Dimes starts the work, funding. We analyze the data. We design the diagnostics with samples off the we design it with test samples at Stanford, UCSF, and all these commercial repositories with the Spark grant. We start a company, raise $2 million in financing. Now let me be crystal clear for this academic audience. I'm not showing you $2 million to brag about it. Actually, it's not a lot of money to raise in Silicon Valley. I'm showing you $2 million because I'm more convinced than ever before this is the new way science will continue out of academia. We got the grants. We got the papers written. If we want to go further, we're going to have to start a company, make a product out of this. But the science still keeps going. We still have to run a multi-center prospective trial. But now that trial is funded on private dollars, $2 million. Or to put it another way, do you know how hard it is to get a brand new $2 million NIH grant today? You know how hard it is. Some of you are nodding here, right? So the idea here is the science continues now in a startup company funded by private dollars. We don't know if this is going to work or not, but we have to do a prospective trial to do it. But because this could become valuable for patients, let's just do this in the company world and innovate that way. That's why I'm showing you $2 million. Now, I love this one. This became a one-man company. Matt Cooper got all the work done by contractors and teams. I'm showing you this one because, believe it or not, last year we've already sold the company. It's already been acquired by a company called Progenity. Some of you know what that means, right? So we went from public data, which people think is as valueless as kitten videos, right? We designed this diagnostic, we test samples off a repository, we spin out a company, raise $2 million, get the trial launched, sell the company, start to finish, 24 months. Inventors happy, investors happy. Are we going to do more of these? You better believe it. And I'm giving away my secrets here, okay? Because such is the need in medicine that every one of you can pick a different disease and we never step on each other's toes. That's how many diagnostics we need today in medicine. Of course, we're going to do more of these. And this is literally how we did it. Everything's described in the papers, how we went start to finish here. And remember, I have no fancy microscope. I don't have a nifty flow cytometer. In fact, my computers are ones I lease from Amazon. It's called the cloud today. I don't even have a cluster anymore. Everything I'm going to show you, I know I admit I make it look easy, but stuff you could learn how to do. There's no special hardware involved here if you want to learn how to do it this way. Let's switch gears and talk about therapeutics for a second. Now, the story for therapeutics is even worse. If I ask even lay Americans, it's amazing lay Americans know this number. How much does it cost to develop a drug? They kind of know. It's $1 billion and it takes 10 years. Now, much of lay America knows nothing in common, but they know this number. It's $1 billion because it's advertised on the Super Bowl, right? It's how you justify pharma pricing in some ways, right? $1 billion, 10 years to develop a drug. Actually, that's an underestimate. It's an underestimate. How do you get the right number? How much does it really cost to develop a drug? Well, it's simple math, okay? Really simple math. Here's the formula. How much did you spend? Divide by how many drugs did you get, right? Pharma R&D spending, divide by the number of drugs. And if you do that math, like Matthew Harper does every year in Forbes, you realize for the top 12 companies, it's between $4 billion and $12 billion per drug. Kind of not sustainable. Now, you may love the pharma industry. You may hate the pharma industry. I'm kind of worried if it's costing this much, where are the drugs actually going to come from, right? Especially for all the other disorders that don't make $1 billion a year for these companies. But actually, let's be a little bit positive. Let's not pick on the pharma industry for a second. Let me frame it a different way. Even if every pharma and biotech company on the planet was 100% successful, there are not enough of them to develop all the drugs we need in medicine today. There's just not enough of them. And then you realize the solution to the problem has to come from us in academia. I love them. I love these pharma companies. I probably consult for them all. You saw my disclosure slide. I don't have faith they're going to solve this problem. It's going to come down to us in academia. We're going to have to figure this out. A more efficient way to get to drugs that we can actually develop. So how am I going to do it? Well, we're going to use data to do this, of course. I'm not saying it's the only way to do it, but we're going to try it with a data problem, and we're going to try our best. And so when we start to collect all these disease samples, disease and healthy, we start to realize that people are running drug experiments and putting that data on the internet. This is Todd Golub and Justin Lamb and others, the Whitehead Institute, Broad Institute, many others now. So they're studying cells and cell lines with a drug, without a drug. So on the left, we got samples with disease, without disease. And on the right, we got cells with a drug, without a drug. They're not even the same cells often. And we're going to put these two together. So the reporters for this call this Match.com for drugs. Okay? Some of you know Match.com, it's how you find a date or maybe a mate or a spouse, right? But what's a famous saying when you're trying to find a lifelong partner? Opposites attract, right? You've heard of that? So that's our methodology here. I'm going to show you all the bioinformatics with my arms. If I've got a disease where this gene goes up and this gene goes down, and I can find a drug now on the other side that can make this one go down and this one go up, maybe there's a match there. It's as naive as that. That's 2 genes. Imagine 20,000 genes. Okay? I'm just looking for a drug that might reverse the molecular profile I'm seeing in the disease. Now, there are 2 C words I'm not saying here. The first C word I'm not saying is cause. I don't think these drugs are going to hit the cause of the disease. I'm not even claiming they're going to, okay? Actually, in medicine, I don't think we have any drugs that hit the cause of a disease. Maybe statins for heart disease, that's about it. Everything else treats what we see. The other C word I'm not mentioning is cure. I'm not claiming these drugs are going to be a cure for the disease. They're going to be a treatment for the disease, okay? That's all I'm expecting out of this. So we're going to turn our crank. We turn our crank on our computer, we got lots of ideas. And here's a chemical, and there's a chemical that might work. And where we got a lot of traction aren't just the new drugs, but the new uses of the old drugs, or drug repositioning. We did not invent that concept. It goes way back. Of course, everyone knows that first cardiac drug that had hair growth as a side effect. That's minoxidil. But of course, everyone knows the other cardiac drug that had its own interesting side effect. Of course, that's Viagra. And of course, in peds, we kind of know Viagra's done a double switch to Revatio. So Revatio's the same molecule, but for pediatric pulmonary hypertension, and people were making fun of babies on Viagra at the time. It opens those blood vessels up as well. But you know what? Instead of finding these by accident, how about we find them on purpose using public big data? And so that's what we did here. Now, by the way, I just committed 2 CME sins in that sentence, okay? I mentioned an off-label use of a drug, so don't go using these things. I mentioned the brand names of a drug. I should never be doing that, right? So if you were into CME, ignore the last minute of what I just said here, right? Because I know those are sins. All right, so we're going to turn our crank, and we got lots of interesting ideas for drugs. That drug might work in another disease, and this one might work in this disease. And so how are we going to test these, right? We got all these computer predictions. How are we going to test them? Well, we got to run a clinical trial. Oh my goodness, before you put this into any patient, you got to show this works in some kind of preclinical model, right? A mouse model or something. And so all my mouse friends and collaborators, they scatter because they know I'm going to give them a lot of work to do. We've had some really great ones in GI and in cancer. But now, when I really, really, really need a mouse model tested, I use something new, which also changed my career. I go to assaydepot.com. Kind of sounds like Home Depot, and you're probably right. Well, let's go browsing in the aisles here, right? Biology, chemistry, DMPK stands for drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology and toxicology. Okay, let's look for a mouse. Let's click on pharmacology here. What type of mouse do you want to buy today? Bone, cardiovascular, dermatology, diabetes, general ear, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I guess I'm a diabetes guy. Let's click on that. Here, I can order a 16-mouse OB-OB model. Maybe I have 2 groups of 8 or 4 groups of 4. Maybe I'm testing various doses of a drug. I send all that drug blinded. What's OB-OB? Well, if you read here, it says this mouse has been eating a lot and getting diabetes since 1951. This is a standard model of American diabetes. You eat a lot, you get type 2 diabetes. You know, it's not the best one, it's not the worst one, it's just one that a lot of people use, okay? And in 28 days, this company will get you the fasting blood sugars, the insulin tolerance test, the glucose tolerance test, all that work done for you in 28 days. Now, what am I covering up here? The price. $9,000 for this service, 9-week turnaround time, and literally, I kid you not, add to shopping cart. This is the new world. You can order an entire mouse model off the internet with a credit card today. I use this like crazy now. Now, I know there are a bunch of mouse people in the room here. I've seen the posters. I walk by them, right? So there are a couple of mouse people here. There's a lot of molecular people here. Some of you are looking at 16 mice for $9,000. That's ridiculously expensive. But that's because you have slaves, graduate students, that can do this work for much cheaper, right? But for dry bench scientists like me, what does MasterCard say? This is priceless. To get this data, right? It's just priceless. $9,000, I'd pay that to get to know if my computer prediction's working, right? Now, some of you with really sharp eyes are noticing this is run in China. Some of you may have a problem with that, others not. I don't. But let's just go further here, just for transparency. When you actually hit add to shopping cart, it actually presents you with the 133 companies here that actually will do this work for money, including Maryland and Wisconsin. So that, by the way, that means outsourcing doesn't mean offshoring. You can pick wherever you want this thing done, right? Whatever company you want. Some of you are super concerned right now because all that computer stuff is kind of neat. But based on this result, you're going into humans next. You're worried about, well, how do you tell the quality of this? Well, there's certifications down here. If I need them to be FDA approved, I can click on FDA. There's 5 of them. ISO 9001, 7, AAA LAC 28, GLP 48. You click on certifications. You only get to see the companies with that certification. You know, I wish it were that easy to pick the right collaborator on campus. That someone with certifications was doing this instead of some random undergrad. I don't know. I wish I had that on my campus. Some of you are still critically concerned, though. How do you trust the results of sending this experiment out and outsourcing this? Because the human study is next. And I will just simply say, if I really, really, really need to know if this mouse model is working, I just order 2 of them from 2 companies. Hell, at $9,000 each, I can order 10 of them from 10 companies. Why am I answering it this way? Think about this for a second. When a mouse researcher gets a mouse experiment to work, do they go across the hall to get a completely independent team of researchers to repeat the experiment? You know, nobody does that. But I can hire as many independent teams as I want, right? To just keep making sure I'm getting the right answer. So I can take that quality problem that's in your minds and now make it a function of dollars. You have more dollars, you can hire more independent teams. Not enough dollars, maybe you can only hire one or two. So we typically will hire one from the West Coast and one from the East Coast. I have nothing against the flyover states. It's kind of easier to do it this way, right? Some in Boston, some in California, and that's typically how we get this done. I can just hire as many teams as I want, and the quality problem disappears. So here's the funny part. I don't trust a single one of these public experiments that where I get the data, but I trust what I get in common. I don't trust a single one of these companies, but I trust what I get in common. I don't have to run the pipetter. I don't even have to touch the mice. And by the way, the amazing thing about outsourcing the mouse models is I can work on inflammatory bowel disease one week and work on cancer the next week because I don't have to spend forever in building the mouse colonies here and nothing, right? I just can switch diseases just like that. Maybe something useful today for precision medicine. So we've done a bunch of these. The first one we did was, we got us in the front page of Wall Street Journal, is we predicted topiramate, a seizure drug we predicted works on inflammatory bowel disease. And I'm not going to show you that because to save time. This is my favorite though. Imipramine, tricyclic antidepressant, we predicted works on small cell lung cancer. So, this is not a pediatric killer, I admit, small cell lung cancer. Humans smoke to get small cell lung cancer, right? You knock out these three genes, this mouse gets small cell lung cancer. This is mouse getting mouse cancer. It's not a xenograft or PDX. You see the nodules here? Mouse gets mouse cancer. We've done a dozen of these, but this is my favorite. Because this is the same mouse treated with imipramine. So what's imipramine? It's a tricyclic antidepressant. It's still in the pharmacy, believe it or not, but we don't use it anymore because we use serotonin reuptake inhibitors like Prozac, right? Imipramine had some side effects. Anyone remember? Makes you sleepy, and it might set you up for an arrhythmia. QT prolongation might set you up for an arrhythmia. Actually, neither of those two side effects sound as bad as having lung cancer. 5% survival rate in 5 years. And the cancer is melted away. It's gone in this mouse here. So why am I showing you this one out of the dozen we've done? We start with public data. Remember, as valueless as kitten videos. We get computer prediction that this drug is going to work. We test cell lines in a dish. I'm not even going to show you that work. This was all work done by Alejandro and Julian there, and Nadine and Joel did this work. We test cell lines, it's working. We get this mouse picture. And based on this picture, 15 months after the computer prediction, we got IRB approval. We launched a clinical trial on this one. Humans are dosed with this drug. Total cost of this trial, $50,000. Not $1 million, not even $100,000. Drug's already there. All you do is pay for physician time and some CT scans, basically. It's a phase 2 AAA study. Is there any biology here at all? Now, I'm financially connected to the molecule, so I can't be part of the trial. It's either going to work or not. It turns out of those two side effects, which one's the worst one? Sleepiness. Turns out if you're a patient that's been told you have 5 months to live, you don't want to spend it sleeping, okay? So we can't even get to the maximum doses on some of these patients. But we got a lamppost, now we got to search for more keys, as the saying goes. I don't care if these work or not, we got to run more of these trials in academia. Nobody is targeting small cell lung cancer right now. And forget about small cell, there's so many other cancers that not a single biotech is actually working on right now. No one's going to do this if we don't do it ourselves in academia. We have got to do this. So we're going to launch a bunch of these. And in Silicon Valley, you got to start a company. So here's the next one. A company called Numeti. We've raised more than $5 million because now there's actually some commercial ability here, right? You got to pay for a trial. I'm not saying to get $1 billion here, but you got to make enough money at the end to pay for a trial. Otherwise, you're just going to end up with more off-label uses. That's not kind of cool, so you got to pay for a trial. And some of you have heard of Allergan. They're in Orange County, right? Until a month ago, they were going to merge with Pfizer and be the largest pharmaceutical company in the world. That kind of fell apart. But Allergan makes Botox and a couple other drugs. This team, Numeti, is about 7 full-time people. And this team of 7 full-time people literally are making the next psoriasis drug for Allergan, right? This tiny little team is now a drug discovery team because they're enabled with 2 million samples and all these mouse models ready to go. So I call these garage biotechs. For some reason in Northern California, we revere garages. I don't know why we love garages so much. Apple and HP started in a garage, so why can't the next Genentech start in a garage or an Amgen, right? That's what I call a garage biotech. I don't mean making meth in your garage. I mean cancer drugs and diabetes and stuff like that. It's all possible. So that's why I love these kind of companies. All right, let's close it out. Where are we going next with all of this? What's the next big open data that's coming next? If you ask, and half my lab is working on this, you know, the saying, skate to where the hockey puck is going. Where do I see the puck going? Clinical trials data. What's a clinical trial? The most expensive experiment in the world. And half of them fail. It's why it's costing us billions, by the way. Half of them fail. We have this weird culture that we don't even write a paper about it when a big clinical trial fails. You know, it's going to change. We never release the data. That's going to change. EMEA, which is the European FDA, is requiring raw clinical trials data release. I don't mean that summary table you got to put on clinicaltrials.gov. I mean the raw data. Every patient, every encounter, every arm, every assignment, every dose, de-identified, of course. All that data I see is going to become publicly available. Now, IOM and NAS are working on this. Pharma obviously doesn't like this. There's some interim solutions there. But I see this world where all that data is going to be publicly available. I run this repository called Import, which we were just mentioning, where we're the only website really in the country now that we give out more than 200 raw clinical trials data, including autoimmune, transplant data, immunology data, rheumatology data. It's really around the immune system and those kind of drug trials. All that data is there. What do I mean by reanalyzing this data? Well, of course, you want to reproduce what the papers had, but I'm also talking about new ways to do meta trials and meta analyses, not just from the p-value level, but actually each patient by patient. Why do you think this drug failed? It seems like it's working in a quarter of the patients. Let's find a marker for that. And those are the kind of papers we've just recently been publishing. I recommend all of you who are interested in clinical trials, just go sign up for a free account. It takes a couple minutes, and then you can download up to 100 of these kind of studies, raw clinical trials. It's a good time to learn about how to do this kind of work. Let's get out of the molecular world for a second and talk about wearables, non-molecular stuff. How many of you have a wearable of any kind? Apple Watch, Fitbit, Withings, Jawbone? Okay, the hands get longer. It's about, let's say, about a fifth of the audience. That's being generous. In Silicon Valley, it's about a quarter usually of the audience. On the East Coast, it's down to 0. So we're still a little bit ahead there in California. Now, this is field of quantified self. You know, it's kind of iffy. Most medical people don't do this, but most lay public people do. It's kind of funny. And we kind of make fun of these folks. I used to make fun of these quantified selfers, right? They're measuring everything that comes in and out of their body. Measuring things that go into your body is tractable. Measuring things that come out of your body sounds gross to me. It's kind of like I's and O's in the ICU. Why would you want to do this at home, right? Why would you ever voluntarily want to do ins and outs? So I used to laugh at these people until December of 2012. I came back from a trip to Hawaii. I stepped on a scale, and I was 247 pounds. And then I realized when I hit 250, they can measure me in tons, right? That's a quarter of a metric ton. It's like, what the hell happened to me after residency, right? I used to walk everywhere in the hospital, driving everywhere, bought everything Fitbit makes, lost 50 pounds, okay, in 2 years. I measure everything. So I've written down everything for the past few years, what I've eaten. It doesn't have to be perfect. It's close enough. It's called the Hawthorne effect. You just measure something, it gets better. I weigh myself every morning. Now, look, I'm in pediatrics. I know what we tell in adolescent medicine, the anorexic kids not to do this, but I do this. I know what we tell them because actually it helps a lot. If I'm even just slightly higher the next day, I can compensate my eating right away, so I don't go off course that much. It actually, day-to-day weight actually does change that much. You have to measure your dry weight. I'm not going to be gross, but you know what that means. And, you know, so, and more importantly, it's a social network. So all of my friends on Facebook know that I've lost my 50 pounds, right? And if you don't believe me, look at my TED Talks. You'll see what I used to look like, what I used to weigh. Now, here's the funny part. So I go to my doctor at Palo Alto Medical Foundation, which is Sutter, which is neither Stanford nor UCSF. It's kind of the competition of both, so it's kind of embarrassing. My own primary care doc is at Sutter. And it's written in the newspaper, they spent $1 billion on Epic. Okay, it's no joke. It's in the newspaper. They spent $1 billion on Epic. But my doctor was the last to know I lost my 50 pounds because their $1 billion system doesn't talk to my $40 gadget. Can you smell the opportunities here, people, right? Let me put it this way. In terms of entrepreneurship, there's tons of companies now working on this. But I'm just going to be a little bit more kind of fear-mongering for a second. If we don't get with the program in medicine, we're going to get cut out of the loop. Because there are many companies that are willing to come in, and they're going to hire doctors like Google and Verily, but they're going to give medical advice this way and get to our patients between encounters. And saying Epic doesn't support this is not an excuse for us in medicine anymore. We've got to connect to our patients between encounters, or we're going to get cut out of the loop. There are many companies that just would love to step in and take over this treasured relationship today. So we've got to get with this program. I'm not saying UCSF is any better, but UCLA, USC, and UCSF all have digital health programs now. I mean, we're all working on this in California, but we got to get better than just the companies and work with these companies. Another source of big data now is cost and charge data. Now, this was funny. I was giving grand rounds at the University of Miami here when this showed up, front page of Washington Post, that they charge double what Jackson Memorial does across the street. Okay, if you've ever been there, you kind of know it's kind of weird. They're literally across the street here. Here's the street. They charge for heart attack with 4 stents $166,000. Jackson Memorial is at $90,000, right? So in some items, it was almost double the price here. So of course I made fun of them during grand rounds. And you know what they said back to me? They said, it's okay, we can charge double than Jackson Memorial because our quality is better. You can't say that when the quality data is public too. Oh, no, you cannot, right? So cost and quality of the delivered care is becoming public. How is the public going to respond? And you know the reporters are better at analyzing our analytics and our finances probably better than we are right now. We cannot keep ignoring this. Nobody wants to be on the extremes here. Everyone wants to be in the middle of the bell curve here, right? But all this data is going to become public with sunshine rules and all this is already public. How is the public going to respond? We better get our act together here as well. And so much more data I have no time to talk about. Microbiomes and ENCODE and cancer genome atlases, of course the EMR data, and all this data is just sitting there waiting for you. A large amount of this is publicly available. Even the private stuff like the EMR data is available to those of us who work in these kinds of institutions here. So where are we going next? Well, I'm really pleased. I'll make a couple plugs for a couple things and talk about what we're doing next for the state of California. This is one thing I'm proud of. So a whole year before President Obama mentioned precision medicine, actually Governor Brown mentioned precision medicine. People don't know this. In the State of the State address, this was in 2014, in his State of the State address, he ended his State of the State address with precision medicine. Now, I'll be the first to admit, I didn't even know Governor Brown gives a State of the State address, but he did. He mentions precision medicine. He launched it. Now, the first year we had $3 million. Even Sacramento calls that budget dust. They're talking about increasing to 10 million. We'll see if it survives right now. Right now, it's on the downside. We'll see if that goes. But the idea is we took this money and we gave it out in pilot grants. So these are UC-wide, academic-wide, and academic industry partnerships. 2 pilots got money for this. And for the governor's office, we're just cataloging across all campuses and biotech parks all the precision medicine efforts in the state of California. So I'm proud of this. I get to run this as a principal investigator, but it's really Jerry Brown who really saw the vision of putting some money into this. We'll see if it continues. And then now I get to build out this new Institute for Computational Health Sciences. It's great. I mean, we're slumming it out in this brand new building in Mission Bay until they build our brand new building in 2019. It'll be opening right here between the brand new hospital. So this is kids, cancer, and women are over here. The other side on Parnassus is medical and surgical. So we have both campuses running now. We're temporarily here until they open this building, which is amazing, right between Illumina and the new hospital. Illumina is a sequencing company. You couldn't buy a better lot for a group like ours than that, as the real estate agents say, location, location, location. So we'll be there in 2019. So that's great. I mean, I get to do a lot in ICHS. We're going to be building the strongest team in the world we can in biomedical data analytics, developing educational plans. I literally have an effort. I want to make sure every single medical, dental, pharmacy, and graduate student at UCSF learns how to write code. Now, it's up to them to say no, but I'm going to offer them a class on how to write code. Because again, if the 7-year-olds are doing this, we better make sure everyone learns. We're going to try to recruit a dozen faculty over the next 10 years to build this out and build our new data assets for precision medicine. Now, what do I mean by that? I loved every day at Stanford for 10 years. Stanford's a great place. But why did I move to UCSF? It's actually, I love UCSF, but I actually moved because of this tiny little thing here called UC Health. Raise your hand if you've ever heard of UC Health. Yeah, so just a couple, right? So most people never know. This is our superpower. Nobody knows what UC Health is, but it's the most amazing concept in the world. So we have this umbrella organization across all 5 University of California medical systems, right? UCSF and UCLA, both of us are top 10, Irvine, Davis, and San Diego. And Riverside is there too, but they don't have a medical system, but they're a medical school. But we got these 5 medical systems. And we have UC Health that actually organizes across all 5 of them. Did you know that if any one of the IRBs approves a study, you can get it approved by IRB Reliance by the other 4? If you have a collaborator, that happens within days to weeks, not weeks to months. Did you know that if you get a contract from like a Pfizer or a Google, you push it up to Office of the President and it's approved site-wide, right? It's automatic. Nobody can scale like we can in University of California because we have this friendship. Technically, we don't compete for patients, right? We don't. You have your patients, we have ours. Maybe at the very tip top of the level, the patient might decide to come to you versus us or vice versa. But in general, we don't compete for patients. Except for, I guess, some one avenue in Orange County where UCLA abuts against UC Irvine. I guess there's a little skirmish there. Other than that, we're not really competing for patients. In other words, this will never happen in Boston because they have to compete for every patient, even within partners. So why did I love moving to UC Health? Because for some amazing reason, they put me in charge of organizing all the medical records for every single patient that's seen by the University of California. At last count, that's 14.1 million. Actually, we're up to 15 million patients get some care in the University of California. And I'm in charge of building the data warehouse that puts all that data in one place. Now, we've had this thing called UC Rex. Show of hands. How many of you have ever heard of or used UC Rex? That's more than most audiences, actually. You guys get a plus for that. UC Rex is this amazing tool. It's been there for 5+ years where we can count how many patients do we have across the system, whether this and that, and this diagnosis codes. But it took a long time to actually get the data on those patients, months to get that data. And instead of taking 6 months, we're talking about making that into 6 microseconds. You've got the core, now let's get the data de-identified. So we're putting that into a centralized facility here. What are we going to do with all this data? Well, this is the boxes. Here are the 5 medical systems, Riverside, put it all together, the clinical raw data from the Epic and all the rest. Go after the high utilizers, figure out why they're costing more in medicine. Readmissions, nobody wants 30-day readmissions anymore, heart failure, all these difficult ones, these difficult kind of groups we can study site-wide. We got these 5 natural experiments, right? All 5 systems think they're doing everything right, but there's some internal variances in care. Let's try to figure out between us and among us what's better, what's not working. What do I see people doing with this data? I have 10 bullets here for all 10 campuses of the University of California. I think community activists will be able to get de-identified versus this, mash it up with Cal EPA data, maybe try to figure out why there are more diseases in certain areas. Transplant patients should be able to get their data across the UC system because they get to care of multiple UCs. A cancer genomicist should be able to get every single cancer that's run across the UC system in terms of exomes and whole genomes. AI researchers at Berkeley or Santa Cruz, right? We have great AI researchers that are not in the medical system that are just desperately trying to get to our data. I think this data, de-identified, of course, will enable a lot of people to do this kind of work. What am I doing with all this data? I'm building maps. I'm going to end with this concept here. I'm building maps of death and disease. Now, that's kind of morbid. We're all going to die someday, technically. Actually, it's illegal to die without a disease. You know that. Some of you filled out death certificates. You got to put something there, right? So it turns out we've used real data on this, and we want to study how do Californians go from health to disease to death, right? Like a Google Maps of death. That's kind of a sad way to think about it. But here's an example of a map. We get patients that come in with alcoholism. They come in a year later with cirrhosis, and a year later they might have liver abscesses. And some of them die here, or some of them die here, or some people go straight this way. Each arrow is like a year, and the thickness means probability, and the squares mean that they died of that disease. So Hannah and Jay have been doing this for us. Hannah's here, and Jay is actually a Hollywood animator. You'll see this in a second, what Jay's doing here. Right, so they get kind of complicated. Here's an example. Here's heart attack. Everyone knows you can die of a heart attack. And everyone knows you can get heart failure, which means the heart's not pumping hard enough. And you can die of that, but then you end up with lung disease, right? The fluid backs up. And a lot of heart attack patients die of septicemia. I kind of never realized that. You die 3 years later with sepsis. Sepsis is a big killer still. So you die that way. Or you go this way, or you kind of go that way. So with maps, we can start to predict what's going to happen next with our patient, right? Statistically, they're showing up here. What do we think is going to happen next? And my plan, when I open that new building in 2019, is we're going to have this room full of monitors, right? And we're going to have our maps of death and disease, kind of like Google Maps. But you know what the problem with Google Maps is? It kind of shows you the traffic, but it actually doesn't show you every car, right? It can't. There's no way it can show you every car there. But in 2019, when we open that new conference room, I'm going to have my maps of death and disease, and I'm going to study and look at every single one of the 14 million patients, moving from disease to disease to disease to death. This is with real data. You can see as people get older, they're getting more diseases, the colors get brighter, more people dying, more squares here. A bunch of them end up with sepsis and then die of that. A lot of people converge on sepsis because it's how a lot of people die. And we're going to plot our 14 million, and we're going to predict what's going to happen in the next 90 days. And we're going to predict what's going to happen in the next year. And we're going to do something about it. And this, to me, is going to be the new definition of an accountable care organization. One that accounts for the care of its 15 million patients. Not just encounter to encounter, but every day. with the wearables and the mobile and the smartphones. This is what I want to build for the University of California. And we're going to get there with all this data if we all work together on this. That's the idea here. All right, there are a lot of trainees here, so I'm going to spend the last 2 minutes. I just added these slides here, okay? A couple things I'll learn along the way for the trainees here. Now, take all this with a grain of salt here. Whereas juniors, we want to be. You know, a lot of you are junior faculty here, right? And I love this term junior faculty, but I also want to caution you. Junior faculty, I love this phrase, junior faculty is only a state of funding. It should never be a state of mind, okay? You should always be a junior faculty member, right? You should be constantly trying to kick your advisor's ass, okay, to be blunt. I love my mentor, Zach Kohani, pediatric endocrinologist, informatician at Boston Children's. He's always trying to scoop me. I'm always trying to scoop him, and we still love going out for drinks together, right? But you should always, don't just do what your mentor says to do more than what your mentor says to do. Don't get into this mindset that I can't do this. Junior faculty just means a funding state. You should always be thinking like you're hungry to do something more. Train as much as you can afford. For some crazy reason, I went back after my PEDS fellowship and got a PhD because my mentors convinced me how to do this because I used a K award to pay for that. Now, not everyone can afford to go back and get a PhD. I didn't even know what an MD PhD was when I entered medical school. But train as much as you can afford. Go back and get a master's degree if you think you need those courses. Set the level of your peers as high as you can. A lot of times we get into a state, oh yeah, my neighboring lab isn't really publishing this year. I'm okay not publishing this year. Those are not your peers, okay? Your peers are in every other pediatric hospital, every other medical center today. Choose your peers carefully and share your peers, who you think your peers are, with your lab and your research group. So they don't kind of get lazy with your neighbors. Think about the whole country. Think about the whole world. In my lab, in lab meeting, we say this all the time. You just had a great idea. 3 people on planet Earth just had the same idea. The rest is execution now, okay? Who's going to get to the paper the first? You determine your future, not NIH. I have a really kind of bone to pick on this one. A lot of people, a lot of fellows say, yeah, I'll go into academia if I end up with a good score on my K award, right? If I get a good score, then I will go into academia. Let me show you my first score on my K award, okay? Now this is back when they went from 100 to 500. You remember 100 was good and 500 is as bad as it could be. And my first score on my K award was 414. They should have triaged it, but this is back before they even triaged this. At that point, if I had that kind of philosophy, I'd never be in academia today. But if you just keep going, you gotta have persistence, you're gonna end up getting them funded, okay? But you cannot let NIH dictate that for that future for you. It's up to you if you wanna stay in academia. So you scrounge, you get fellowships, you get foundations, and eventually you get the NIH funding. You'll see how much I'm funded at the end. Innovate beyond your university. My wife taught me this amazing thing before we got married. We've been married for 15 years now. Writing the best grant today often resembles writing the best business plan. Because what does that mean? If you're not going to change the world in 3 years with that money, man, you'll neither convince a committee in Sand Hill Road, nor will you convince a committee in Bethesda to give you money to execute on your ideas. I've served on study section for 17 years now. The first thing they drill in your head is significance, significance, significance. Even if they're technically correct, is this going to change the world or not? If not, you're not going to get the money. So think about now, not always writing a grant, but sometimes starting a company. Even in the University of California system, they let us spend 384 hours a year working in companies, actually, above the table. Okay, that's allowed. No one said you have to go consult. You can start your own companies during that time. A lot of people don't. It's the same problem today. If you have a good idea, it's just about getting people to give you money to execute. Who cares if it's a private versus a grant at this point? And shoot for changing the world. Go for real world big problems. There's so many of them out there. Don't believe it when people say they're not addressable. Address the big real world needs, not just the easy ones. There are plenty of people running easy ones and easy abstracts. Go after the hard stuff. The public is depending on us to get to those solutions. So in the end, what is big data and biomedicine? It's about algorithms, programmers, databases, cloud computing, mobile. Yeah, it's kind of about all of these. I didn't show you any of this, right? To me, it's about predicting diseases before they strike, explaining rare diseases before that defy experts, finding drugs for diseases that lack attention, and not enough pharma and biotech companies on the planet to help us, making sure we do the right, safe, cost-effective thing for patients. And to me, this is an amazing platform today for biomedical innovation entrepreneurship. I sum all that up with one word: hope. We have all this data waiting for you. All that data, it's going to be up to you to use that data to get to drugs and diagnostics. My lab is only as big as it's going to get at this point. I need more people to realize that there's so much value there. And you can develop diagnostic therapeutics to really bring hope to all of these patients and families. I have to thank a lot of people for all this work. I have 40 people across the entire University of California system building this data warehouse of the 14, 15 million patients. I don't color code these now anymore based on campus because we're all one campus. We are the University of California, just different zip codes. Okay, so I'm not going to color code based on who's on which campus anymore. A lot of collaborators. I would never get all this biology done without them. I showed you especially work from Alejandro and Julian with the mouse model for the cancer, and Jay Peshek and his team helped us with GI diseases. A lot of others here that we've been working with at UCSF and at Stanford and many other places. And I have to thank endowment support from UCSF, of course, to recruit me a year ago. My lab has been blessed with 17 NIH grants from these 9 institutes of NIH. These 4 give me more, these 5 give me less, but I still love them. Marcia Gimes, HDRF, HB, HHMI, stem cell money. We get disease-specific money to work on specific diseases from foundations. CTSI seed grants at Stanford, UCSF. I always thank my admin and tech staff. I'd never get a grant or paper out the door without them, especially Mary, my admin. I thank Zach for being my mentor and friend for life. Keith and Sam for recruiting me to UCSF. I always thank my daughter. She's great. She keeps me interested in science, and I keep her interested in science. It's important for us to keep young women in science, so she's my challenge with that. She's a science nerd and happy about that, and I want to keep it that way. And I always thank my wife, Darlene Deshpande, molecular biologist, biochemist. Sometimes she collaborates with my lab. She reads every major paper and grant that comes out of my lab. She starts companies with me now and lets me go all over the world to give talks like this. Thank you very much. I think I went over. Is there time for questions?
Amazing. If there are questions. Are there questions?
Sure. Go ahead.
Go ahead.
There's a microphone coming to you.
Thank you so much. This is great, and we learned a lot. I want to ask your advice regarding negative data. I always think negative data is data as well, and it may help us to understand the internal variation and internal diversity, especially that we are in California, are privileged with having patients of different ethnic background.
Yeah, so negative data is great. I think we have got to get out of this habit of not publishing that. At least let the raw data out so others can intersect it, or you can intersect that data with others and figure out what's the positive there. And now, there are journals now that are taking negative data. There's PLOS One has always been taking negative data if you show what you've done. So they're journals, so I encourage you to put that data out there. But put it out in raw form. You'd be amazed. Another data scientist will come and cite your paper if they use your data. So it's not valueless at all. Yeah, question in front, sure. I'll repeat the question, go ahead.
Great, thank you, it's wonderful talking. Using big data is also only as good as the data we put in.
That's right.
And certainly, notoriously, I'm one of them. I get lazy and I don't really go down that ICD-10 code and identify that it's the left foot and not the right foot or
Right.
the front toe. And so it seems to me that we aren't really there, that there's so many errors that can
Right.
occur.
Yeah, so usually this is the first question, I guess. How do you trust the quality of any of this data, whether it's clinical or molecular here, right? So first of all, for the clinical record for a second, a lot of clinicians say actually, how do you trust this data? They remember what they put in there. By the way, I think the EMR data is the most expensive data in America because we pay physicians to enter this stuff in, right? So that I think it's the most expensive data in the United States. So, they might not pay you to do it, right? Because you're doing it at home. You're actually right. So actually, I have a lot of faith in that data, much more faith probably than most physicians do. Remember, I might have a sick patient crashing in front of me on a gurney, and I can legally and legitimately print out that record, put it on the gurney, transfer the patient, and someone else can actually save the life of that patient based on that record, okay? So it's actually good enough today to actually save a life. Of course, we know there's holes in the data. We know there's noise in the data. It's amazing. In America, we have these extremely short, fat people. That's because the height and the weights are mixed up. We know that. We're going to fix those, right? People put heights where the weights and weights where the heights. We know that. We can fix all that noise. And as we use this data, of course, it's going to get better. But here's my more general answer. The data in any form always has problems. And the challenge is that we're trained as skeptics, as scientists. We're trained not to trust other people's data. That's how you're trained in science, technically, right? But the problem is this, is that the data is amazing. So first of all, most data, because when data is doubling, let's say data is doubling every 2 years in some of these repositories, What that means is that when you go to that database to go look at the data, half of the data in the database is actually 2 years old or younger, right? So anytime you go to these repositories that are growing exponentially, half of the data is fresh data. So it's not ancient old data, it's actually fresh data. Most people know what they're doing today. I mean, it's a pretty good data. But here's the general answer here. The problem in science is that we're skeptics. And I will just challenge you to think about this. That you know we're such good skeptics that 50 years from now, we'll still find a way to criticize data collected 49 years from now, right? Why would we ever think data is perfect at that point? You know we won't. So we will always find a way to criticize data, right? So stop thinking that way, right? My whole first few papers in my career were kind of making fun of these microarrays. This one doesn't match that one. And look, the AFI version 4 doesn't match AFI version 5. I used to write these kind of papers, and they're kind of meaningless papers. And today what the game is not about criticizing the data, but just to think, what can you do with this data in its current form? There's always going to be more of it, and it's always getting better. But what can you do with it today? Don't wait for perfection. Voltaire, perfection's enemy of the good here. What can you do with it today? That's the way I think about this data today. I know it's got noise, but I got so many people now entering this data, there's wisdom in the crowd here. That's the idea. Sure.
So I thought this was a fantastic
Thank you.
talk, and I love your vision of academics. It's certainly something I've thought about a lot. I'd be very interested in your thoughts on paying our athletes, because I think that's the same problem, except in athletics, where they make a lot more money than we do, by the way, but I'm not going to ask that. I'm the academic officer.
We don't have athletes. We don't even have a mascot on my campus, so I plead the fifth here. Okay, go on.
I'm the academic officer for our Department of Pediatrics.
Surely.
And one of the things we face is we start to accept the fact that we probably should pay our athletes and we should allow our professors to form businesses and all the rest, is how to evaluate their academic advancement.
Absolutely.
I detect a hint, more than a hint of academics under that veneer of yours. And I'm just wondering, because I'm sure you've thought about this.
Yeah, absolutely. I think a lot about this.
How do you do it for your people? How should we be doing it?
Yeah. So first of all, before you start companies, you have to get your academic house in order. You got to write papers. You got to get grants. Of course, if that doesn't work, if your day job isn't really working, you don't have the luxury to do the rest of this. But when you have that working, you should do more, is what I would really argue. So of course, tenure committees, I mean, even whether it's Stanford or UCSF, I mean, if you start a $1 billion drug company, it's your drug, no one's not going to give you tenure for that. I mean, you probably wrote a bunch of papers along the way. I mean, you're not going to, fail that way, but you got to get that side of things going too. Most of the companies we're talking about are in biomedicine here, so actually human relevant here. But I'm not saying this to the exclusion of actually being a good academician too, okay? You got to get the papers out, you got to get the grants out. But in some ways, that part can be more mechanized. We can be more efficient about that. You got to write a bunch of grants. I mean, that's just the NIH way now. You got to just, in the beginning, write something for every cycle. And then eventually they all get funded, right? You just have to get, writing papers becomes a machine. It's just, I think we publish a paper every 16 days in my lab now. Every 16 days we have a paper published. It's just a machine. We just know how to get these done. Everything's about papers now, right? And everyone wants to write a paper.
Last question.
In the back.
Can you talk about how you see these clinical and biochemical data being leveraged for thinking about health disparities
Great
and
question.
Sort of the social determinants of health, population health?
Absolutely. Okay, so social determinants of health. So how can you use this kind of data? We had this amazing, one of my favorite papers, came out PLOS One. It's already been cited like 300 times, PLOS One paper, where we wanted to use this whole GWAS approach, for genetics. We wanted to do this on the environment. So environment, adversity is typically studied by epidemiologists, right? And so they have this kind of mindset to just study one environmental factor at a time. Let's study this effect or that effect. But the geneticists don't do this at all, right? They study the entire genome to look for an effect. So we said, why don't we pretend we're geneticists but study environmental data this kind of way? So to make a long story short, we wrote a paper called an EWAS, an Environment-Wide Association Study. What data did we use is NHANES, right? National Health and Nutritional Survey. 50 plus years. We all know NHANES because they made the growth charts. But every other year, they go to random cities and towns in America and they measure blood tests in up to 10,000 people. All that data is on the internet. pollutant levels, lead levels, all of that. It's actually all on the internet. So we use that like crazy. Now, this clinical data that we're building across the University of California, why is it going to be amazing? Because we see the diversity in California, right? And in fact, our diversity is the diversity of the world. Everyone comes here. So in some ways, this clinical repository is something no one else is really going to get to, I think. I love Iceland, but that's Iceland. I love Geisinger in Pennsylvania. Even they call themselves the Iceland of Pennsylvania, right? I mean, so those are nice, but we're America. We're the entire world here. So that's exactly why we're going to do this within the University of California. I think we got to call it quits here. Thank you very much.
So our next speaker is Carol Mangione, and I have a great pleasure of introducing her. When we sat around the table thinking about who the speaker should be following Atul, we thought we should have a health services clinical researcher and somebody who's a true scientist and a scholar, and of course we thought of Carol. So let me just do a very brief introduction of Carol. She is the Barbara and Gerald Levy Endowed Chair and Professor of Medicine at our medical school. She's Professor of Public Health at the Fielding School of Public Health. In addition, she's the Director of the UCLA Drew Resource Center for Minority Aging and Research, or RICMAR as many people call it. She co-directs the RWJ Foundation Clinical Scholars Program. In fact, far more than that. I think she, more than anybody, has saved the RWJ Clinical Scholars Program across the United States. But that's another story. She's Associate Director for the UCLA CTSI and directs the educational components of the CTSI. She's also a practicing primary care physician at the Faculty Practice Group, and her expertise includes a number of key areas in medicine: diabetes, diabetes prevention, health disparities, aging, public health, health insurance design, and so on. I've gotten to know Carol a little bit, especially through the CTSI. And to me, the definition of a scholar is somebody who not only does great scientific work, but who lives through her protégés, whose greatest enjoyment is the impact that her protégés can have. And so it's a great pleasure to introduce Carol Mangione, a true scholar.
Thank you for that incredibly kind introduction. And it's the other Mangione who's the pediatrician. So it's good to be here as an internal medicine doc talking to you. And Atul's kind of a hard act to follow, but I think that in some respects, his talk sets the stage for my talk. So my talk is about one of the first disease-specific health insurance plans in the country. And so, as we think about precision medicine and personalized medicine, this is an interesting attempt by a national health plan to actually tailor a benefit to what patients need and to build in financial incentives to make it easier for patients to do the right thing in terms of the evidence-based care practices that they should be pursuing. So we were interested in finding out whether that would work. I don't have any conflicts of interest. So let's think a little bit first about diabetes. So on this side of the slide is the good news, right? So all of the really terrible complications from diabetes are going down over time. And we have strong national data from many sources to support that. Here's the bad news, and those of you in pediatrics know this story well, and that's that the incidence of diabetes is going up, and not only is it going up, but the onset is much younger than it used to be. So when we take these two curves together, even though we've made progress in treating the end-stage complications, so many more people in the population are going to be having them if we can't stem the epidemic in childhood and young adulthood. So what can we do to take care of patients with diabetes? Well, we have a treatment model that's well-grounded in a lot of clinical trial data about glycemic control, blood pressure control, lipid management, and the rest of these things. And then, we also have a prevention model. So we have diabetes education. We know from the diabetes prevention program that physical activity is important. We know that reducing tobacco is important and a healthy diet is important, right? So we have these two approaches, and intervening between the two are a lot of population-based targets. So these are really interventions and policies that help us through benefit design, system design, and community policies and laws to try to encourage the population and the physicians to do the right things with the patients. So CDC got together in 2010 and developed this program called Natural Experiments in Translation for Diabetes, or the NEXT-D program. And the NEXT-D program was a multi-center research network to test the effectiveness of policy-targeted health policies on diabetes prevention, control, and health inequalities. And this whole program was really started at CDC and then in partnership with NIH to try to provide policymakers with clear prioritization of best practices that could be implemented by health plans, communities, legislatures, and government. These were the original NEXT-D teams, and we just joined the second cycle. But these were the first cycle teams, and the study that I'm going to talk to you about today was in the first cycle. So the team at Harvard was studying the impact of high deductible health plans on diabetes care. There's been a massive proliferation in the market of high deductible plans. The team at Northwestern University evaluated the diabetes prevention program and YMCA national partnership. At St. Luke's Columbia, there was an evaluation of an enhanced electronic health record in safety net clinics to try to help doctors and patients do the right thing. Kaiser Permanente came in with a number of very interesting interventions that were kind of light touch, but affected a lot of people. And then we came in with the evaluation of the diabetes health plan. So what is this enhanced benefit design? Basically, the principle of the diabetes health plan is that it decreases out-of-pocket costs for medicines and preventive care. It makes health promotion free, and it has a number of nice compliance features to help patients do the right thing. This health plan is sold to large employers in the country, and it also has the flexibility that the employer group that buys the health plan can tailor it to their employees. So this benefit has been available since 2009. And just to give you a sense of how it's different than a standard plan, because much of what I'm going to show you are comparisons with patients who have standard plans. Primary care visits are free. Drug copays are free for the drugs that people with diabetes should be taking. So glycemic meds, insulin, statins, ACE inhibitors, ARB inhibitors, or ARBs are all free. In a standard health plan, people would be paying at least $5 to $15 per prescription, and for the branded drugs, even more than that. In both the standard plans for most people with diabetes and in the diabetes health plan, lab tests tend to be covered. So if you want to learn more about our design for how we set up this study, we do have this publication, and I really want to take a moment here and acknowledge my co-authors. Kenrick Duru is somebody who has worked with me for over a decade, came in as a first-year faculty member. I also have worked for a very long time with Susan Ettner, a health economist in our division, and I'll be telling you more about some of the other people on our team. So in the next few minutes, I'm going to run you through 4 main analyses that we've published from this work. So one of our first questions was, if you were in an employer group that had the diabetes health plan and you had pre-diabetes, was your probability for transitioning to diabetes less, the same, or greater? If you were in a plan where metformin was free, were you more likely to use it to prevent diabetes? If all your drugs were free that you should be taking, were you more likely to adhere to them? And then finally, if you're in the diabetes health plan, are you less likely to use the emergency room or get hospitalized since all of your primary care is free? So this is the setup for all of these analyses. So let me take a moment and walk you through this. So, when you want to look at something like a health plan benefit, you rarely have the luxury of randomizing. So this puts us into the world of quasi-experimental designs. And what we try to do is we try to find comparable patients in comparable employer groups who have a standard benefit instead of the diabetes health plan, and then we follow longitudinally to see how patients do. So this schema is sort of how the whole analysis is set up. So we have patients with prediabetes and diabetes, and we have employer groups where the patients are eligible for the diabetes health plan. It's offered by their employer group. And then there are employer groups that have the standard benefit, and they don't offer the DHP. For all of the patients in all of these analyses, we have a year of big data, pre-data, and then we have 4 years of post-data. And what we did was we included all patients eligible for the DHP in all analyses in order to really approximate the impact in the real world. So if you had an employer group, like one of our employer groups, the City of New Orleans bought this for their employees, everybody was auto-enrolled. But then patients had the option of opting out if they wanted to have a standard benefit. So when we analyze at the employer level because it's at the employer's decision about what type of health insurance to buy for their population of patients. So we're not just looking at the people who actually got exposed to the benefit. We're looking at this at the employer level, and we do that for all of these analyses. So for most of the analyses that you're going to see, the employer is actually the unit of analysis, and then we use employer-level propensity score matching to find comparable employer groups with the standard benefit. And then a lot of the measures that you're going to see are actually measured at the patient level. So the first question, will the diabetes health plan prevent the transition to diabetes? And this work has been presented in abstract form at a couple of national meetings, and the manuscript is currently under review. So we know that 1 in 3 adults have prediabetes, and that 15 to 30% will transition to diabetes in 5 years. So prediabetes is a really important public concern, and prevention is important. So in this analysis, we looked retrospectively at that year before the diabetes health plan, and we used an intent to treat analysis at the employer level. And what we have here is we have our DHP group, and they have a pre-perioded 2010, and then we have our post data. And then what we did with the control population is we kind of artificially defined a pre-period at the same time temporally, okay? Because obviously the controls have had a standard benefit for longer than just 2010. In our match, we included employer size, salary, age, percent female, race, ethnicity, percent with chronic conditions, percent with prediabetes, and then you can see the other factors in the model. For our primary outcome, we defined it as diabetes diagnosis over the 3 years among patients in the employer groups who had prediabetes. And for those of you who care about the details, this is how we actually did the definition based on claims and labs. We then had the primary predictors. If you think about the treatment in this analysis, it's really whether you're in a DHP employer group. And we used multiple imputation for missing demographic and lab data. Last talk talked about, a little bit about missing data and dirty data. That's a fact of life in this field. But there are some good statistical methods for filling in the missingness. And this becomes critical in our field. Most of us could not get papers published if we didn't use statistical algorithms to deal with missingness, because the complete case analysis is often a very biased sample. They're the most compliant people. The people come to the doctors the most. They get the most lab tests. And information on them may not generalize to the population at large very well. So how did we do with our propensity match? Well, you can see here that in the DHP groups, we had more females. Our age match was pretty close, though our DHP patients are a little bit older. We had a slightly different racial and ethnic breakdown with basically more African Americans in the DHP group than in the control population, and fewer Asians. We also had a slightly less educated group in the DHP group, and we had slightly lower income. So what did we find? We found that the predicted probability across the 4-year follow-up time for people who were not in a DHP employer group was 37%. For patients who were in the DHP employer groups, progressing on to diabetes happened 29% of the time. So we saw an absolute difference in progression of 7.6%. This is a really big effect relative to a lot of the things that we can do to prevent diabetes. We were interested in knowing more about who were those patients who had the highest probability of progressing on to diabetes. And I think what we found isn't that surprising. Older people were more likely to progress on to diabetes than younger people in the sample. People who started out with a higher A1C, so they were closer to the threshold, were more likely to progress on. And people who, by ICD-9 code, had obesity were more likely to progress on to diabetes. So our analysis showed that the risk of progression from prediabetes to diabetes was significantly lower for those in the DHP employer groups. And so benefit designs that reduce cost sharing for preventive care, lifestyle modifications, and use of metformin seem to delay onset of diabetes. So many of you are probably familiar with the diabetes prevention program. And to me, it's a really interesting clinical trial. So it published in 2002 in the New England Journal. And what the diabetes prevention program showed was that a lifestyle intervention where patients achieved about a 7% loss of weight and where they exercised equivalent of 30 minutes of brisk walking 5 days a week reduced the risk of diabetes very substantially. Now, in the diabetes prevention program, this was a very high-risk population that they selected out. It was a multi-ethnic population. It was one of the first times the NIDDK really made a huge effort to make sure there were enough Hispanics and African Americans in the sample to be able to look at those groups. But what a lot of people forget is there was another arm in the diabetes prevention program, and that was the metformin arm. So, you know, in our lab, we always talk about how, for some reason, metformin just got lost in translation, right? So here, you know, if you go back and look at those original New England Journal papers, the patients who were randomized to metformin had a 38% reduction in proceeding on to diabetes in a 3.5-year time window. So, you know, let's compare that to a lot of the therapeutics we use today. If we think about the effect of statins on reducing 10-year MI rates, it's about 30%, right? So we have a lot of therapeutics out there that, you know, have a similar effect size, but for some reason, uptake of metformin seems like it's been really low. So we wanted to provide some of the first empiric data on what the uptake actually was nationally in this large insured sample. So this work was led by Tanaz Moyen, who I want to acknowledge, the first author here. Tanaz is an assistant professor, endocrinologist here at UCLA and at the VA. Tanaz boarded in endocrine and then did a 2-year health services research fellowship. So she has really unique training and is really quite phenomenal at doing this work. So as I mentioned, in 2002, the DPP showed both metformin and lifestyle delayed onset of diabetes. Now, in 2008, the ADA care guidelines added use of metformin to prevent diabetes. So we have a lot of long-term safety, tolerability, and cost-effectiveness data, but little was known about the uptake in the real world. So what we wanted to do was characterize the rates of metformin use among insured working-age adults with prediabetes, and to test the association between patient characteristics and predicted probability of metformin use. So in this analysis, we did a retrospective analysis of administrative claims data from 2009 to 2012, and we used the prediabetes diagnostic codes in 2010. We excluded patients with polycystic ovary syndrome since they might have another indication for using metformin. We excluded anybody who already had diabetes or had a history of gestational diabetes. And what we found was that if you used among these 17,000 working age adults that came from all 50 states with at least one prediabetes code. So that means some doctors somewhere thought they had prediabetes. And among that group, we see a rate of 3.7% for any metformin prescription. So who was more likely to get a metformin prescription? Women were slightly more likely at 4.8%. People with a high BMI were more likely at 6.6%, and people with more comorbidity were more likely to be on metformin. So we concluded that metformin was rarely used in the management of prediabetes, despite strong evidence from randomized controlled trials for its use over 10 years. One of the barriers may be that it's an off-label use. So the FDA has never approved metformin for use in prediabetes. Well, why is that? Well, it's probably because of the economics. So metformin costs about 4 cents per tablet. It's available widely in generic form. And to get through the FDA is a really expensive process to change your labeling, and there isn't really a branded pharmaceutical that's going to gain any profit from doing that. So a number of scientists in the country, including myself, have formed a citizens petition to the FDA based on this paper and based on the work of one other group at Kaiser to basically ask them to change the labeling on metformin so that it can be used for prediabetes. So this has been an interesting process, and it's where we've taken really, our data and our publications and have tried to leverage it to affect policy nationally. And I think many of us health services researchers, that's really our goal, is to really have these types of findings inform federal policy. So what about the Diabetes Health Plan and medication adherence? And this analysis was led by Kenrick Duru. We also had a wonderful collaborator up at Kaiser, Northern California. Romaine is an incredible modeler and statistician who helped us with some very thorny issues with conducting this analysis. A lot of these other authors here are actually from United Healthcare. And United Healthcare has been a partner in my research and has provided me with access to data for over a decade. The data in all of these analyses, though, reside in our shop at UCLA. We get PHI-compliant limited data sets from United. We conduct all of the analyses, but we also talk to them on the phone once a week for an hour. We have a standing conference call, and that becomes really critical because these data are very complex, and oftentimes the people in the actuary groups can help us understand what we have. And so that close partnership with the health plan has been critical for this work. So why medication adherence among people with diabetes? Well, we know that glycemic control is important. I don't have to tell pediatricians this, but if you decrease A1C on average from 8 to 7%, you can reduce microvascular complications by 25%. We know that if you can control blood pressure, you can reduce complications, and we know that lipid control is critical, and I've cited some of the big trials here at the bottom of the slide to support these three observations. So our specific aim was to determine whether the effect of an employer purchase of the diabetes health plan changed adherence for metformin use, statin use, ACE, and ARBs. Again, why an employer level analysis? This really is because, from a policy perspective, employers have to decide whether to purchase the DHP and offer it to their employers. So this is very much set up analytically like an intention to treat analysis. So we have employer-level propensity score model to identify control employer groups most comparable to our DHP groups. And then we use a claims-based algorithm for the proportion of days covered to assess employer-level medication adherence among all people with diabetes over a 9-month period. So how does this work? This is another great way to leverage claims data. So it's widely accepted that you can use pharmacy claims and the amount of drug that people purchase to estimate their adherence. So for this analysis, we consider adherence to be good if somebody has purchased enough pills that they would be taking the medication 80% of the time. Okay, so that may not sound so good to you, but the average adherence nationally for patients on chronic therapies is about 50%. And so among the health services researchers who use claims data to assess adherence, the benchmark of 80% has been kind of a widely accepted criteria. And then what we did was we used an inverse probability weighting to determine the average treatment effect on the treated. So in this case, the treatment is being in an employer group with the DHP. So what the average effect on the treatment on the treated is the difference in predicted adherence among employer groups who purchased the DHP and the predicted rate had they not purchased the DHP. So that out of employer group prediction comes from the controls, okay? And so the treatment, again, is the employer group purchasing the DHP. And the differences that I'll show you are the predicted rates with purchase of the DHP compared to the predicted rates had those same employer groups not predicted or purchased the DHP. So what did we find? We found basically a significant but modest effect. So for metformin, people with the DHP, 67% of the patients were in that above 80% adherence group compared to 63% with those with standard benefit. Statin use was about 5% higher, showing a 5% higher rate of being highly adherent. Same with ACE and ARBs. Now, you could be concerned that maybe there's something special about these DHP groups. So another thing we oftentimes do in health services research is we'll try to look at something that shouldn't be affected by the benefit that we're interested in. So what we did was we picked a rather expensive at the time asthma drug, and this didn't have any cost reduction in the DHP compared to standard benefit. And we wanted to see whether there would be an impact. And we were reassured to see that basically this difference is just below statistical significance, but it's in the opposite direction, suggesting what we're attributing to the benefit design may truly be from the benefit design. So we found differences of 4 to 5 percentage points in medication adherence at the employer level attributable to the disease-specific benefit design, and that the combination of free and reduced cost medications may be a promising approach to improve adherence, but clearly we need to do a lot more, right? So my final analysis that I'm going to show you is the impact of the diabetes health plan on emergency room and inpatient utilization. So why focus on emergency rooms and hospitalizations? Well, about 26% of all hospital days and 12% of all emergency room use is incurred by patients with diabetes. So this is really a big ticket item for us all nationally. So we wanted to compare annual adjusted rates of emergency room and hospital use for employers who purchased the DHP with rates for those same employers had they not purchased the DHP. So the same setup as the last analysis. We used an employer-level propensity score match, and we examined data from 3 continuous years of data, and you can see the criteria for the specific employees that we included. And again, we used an inverse probability weighting to determine the average treatment effect on the treated in this analysis. And what we found was in the 1 year post, the predicted emergency room use without the DHP was 18.9%. And in the 1 year post with the DHP was 16.5. So that's a 2.5% reduction in emergency room use, which may not seem like much, but if you take it relative, it's about a 13% reduction and it's statistically significant. At 2 years, we saw a little bit of a decline, but a comparable level statistically in terms of the reduction in emergency room use. So patients who had free access to their primary care docs did seem to be using the emergency room less. Our hospitalization data was not significant. Now, that's probably due to our sample size because these are relatively healthy people and hospitalizations were relatively rare events in the sample. So in summary, our analysis showed statistically significant reduction in adjusted mean rates for emergency room use at 1 and 2 years post the diabetes health plan, and no impact on hospitalization. So to wrap it up, the diabetes health plan is associated with improvements in several outcomes that we care about as doctors, patients, and society. So patients with prediabetes showed slower progression onto diabetes, and patients with diabetes also were shown to have better adherence to the treatment once they crossed those thresholds, and we also saw lower use of emergency room services. So at the same time, the DHP didn't lead to people with prediabetes using a lot more metformin than those with the standard benefit. The rates were actually comparable. I didn't show you that, but I just showed you the overall national rates. And, you know, doing this work partnered with United Healthcare has actually been very important, because while we're busily doing our modeling and presenting at national meetings and getting our peer-reviewed publications, they use our information right away, and they start tailoring and changing their benefit. And because of what we found in this study, they actually have now, in the next 5 years, they've built in financial incentives for patients to pursue lifestyle. So online lifestyle was free. Now they're going to pay people to do lifestyle interventions because they saw the very low rates of uptake from our data. The other thing is they've seen that the offset in emergency room use by far and away covers the cost of the free drugs. So the idea that they might expand the pool of medications that patients need and make them free works financially for capitated employer groups, because they're going to see a lot less spending on emergency rooms. And, you know, I think most patients would probably prefer to take free drugs and not end up in the emergency room. So it's probably a win-win all around. So, you know, what do we walk away from this? I mean, this is one chronic condition, one tailored benefit. But you know, it really shows us that condition-specific health plans may enable focused care delivery and better results for high-risk populations. So we really see this as a model. So now take this to the big data environment. And you know, with the EMR data, we actually have the very possibility of not just tailoring a benefit for something like diabetes, but really having employers and health plans think about all of the conditions that their patients have, and think about, you know, which drugs should really be low cost or free for which patients. You know, which therapeutic strategy should be low cost or free for which patients. So, you know, if you want to decrease your rates of unnecessary back surgery, maybe you should lower the threshold for the non-surgical interventions for the people with chronic back pain. You know, and so one could imagine that you could actually take this model and expand it out to really tailor a benefit to what people need. So we're in the process of negotiating to continue to follow the diabetes health plan patients for 10 years instead of 5. And we also have some global cost and cost of medication analyses underway that will be presented at the health economics meetings in June. So I'll stop there and acknowledge my huge list of collaborators, both at UCLA and in United Healthcare. Thank you.
Thank you, Carol. One of the great benefits, that was a great talk. One of the great benefits of being UCLA is that we could learn from different disciplines. And so as I was listening to you, I've done a lot of work in health insurance plans, but I've never really heard about a condition-specific benefit package, and I could easily see applying this to childhood asthma or ADHD or mental health conditions.
Yeah.
So we do have some time for questions. Yeah, go ahead, Paul.
Hi, Paul.
That was great. That was amazing. Thanks. Just one question about the research, and then a broader question, just sort of about how to apply this to other issues. But was there any evidence that the metformin and the ACE and ARB and what was the third one, and the statins actually mediated some of the ED effect?
Yeah, so, you know, I think the mediation question is really complicated, and it's hard because we really our statisticians have really tried hard to keep us at an employer level analysis. And there are a lot of statistical reasons, given the way this benefit is distributed in employer groups and around the country, why that's important. But, I think that's a really key question. The one thing we have been able to look at is, are the people who are not going to the emergency room actually using primary care more? And they seem to be. But again, that's also a very tricky kind of endogenous problem too, because sicker people see primary care more, So it's that's what we would all like to know. And again, I think when we think about the future and big data and being able to link EMR data to benefit design, like at a system level, you can really begin to explore those relationships a lot better. So I think the previous talk about UC Rex and the potential, UC Rex also does have all the health insurance data in it, and a lot of the other aspects of care that one might want to examine.
So that actually leads into that second question, which was the bigger idea of of using UC Rex and using big data and using the UC Health systems data. And I mean, one of the things that was sort of unstated sort of throughout the talk was just the fact that when you go back and you look retrospectively, that nothing works unless the propensity modeling works to a certain degree.
Yeah.
And so for all of us, when we're all now thinking about, okay, so what are these condition-specific health plans? Like, is it going to be asthma? Is it going to be this? But for us to actually use UC Rex and to use the UC Health System data, or to partner with a health group and use their retrospective data, we need to be confident about that aspect of it. And
we've got propensity score models with UC Rex data for other studies, not the one I presented to you today. This was United. This is 50-state national sample. But we've been another book of work that I'm doing embeds clinical pharmacists into primary care, and we're really interested in seeing what the impact of that is on people with chronic conditions. And so, for that study, all of our propensity score modeling is in UCLA Health data. And I think we can do a much tighter patient level model when we're using UC Health data, because we've got the EMR. We're not just in the world of claims. So for these analyses, we had almost 100% capture of pharmacy data, utilization data, very strong demographics, lab data on about 35% of this sample. So for our lab endpoints, which I didn't focus on today, we've had to do a lot of imputation and out of sample estimation. So, but when we're using EHR data with benefit data connected together, then, we're going to have much more complete data. So that's an exciting thing. And I think also, I sit on the UC Healthcare Task Force for the 10 campuses, and we really have three predominant plans, right? I mean, we have HealthNet, we have UC Care, which is UC's first attempt at self-insuring, and then we have a large population of employees at Kaiser. So, this idea that we might be able to use UC Rex to look at variation in benefit design and link it to longitudinal outcomes for children or adults with chronic condition, it's like there's no reason why we couldn't do that. So.
Oh, that was very good, Carolyn. Like Paul, I tend to study the government publicly insured group. So the employer insured group is an interesting group to me. But it seems to me, I'm just curious if this came up, employers also have a interest in this, because if their employees don't go to the emergency room, it means they're at work, or they're not missing days of work, they're not missing productivity. And I was wondering if, first of all, if you were able to study that at all as an added benefit of what you're doing, but also whether the employers who chose the plan actually knew that perhaps they had a higher incidence of diabetes in their population. I mean, there's some things in your data that suggest they might have, and how are they making that decision?
Right. So, the employer for United is the customer. Okay. And we actually, they're kind of hands off about letting us talk to the customers. But what they did let us do was some qualitative interviews with the sales reps who do talk directly with the customers. And it is clear that employer groups who, they kind of broke down into two groups. Some have vision about keeping their populations healthy, right? So one of our employer groups is Hewlett-Packard. Well, you can't compare Hewlett-Packard to City of New Orleans, right? And so we compared Hewlett-Packard to another Silicon Valley company that had a standard benefit, okay? So there are some companies that just have a culture of wellness, and so they're looking for benefit designs like this because it fosters kind of their whole way they think about the health of their employees, okay? And then you have, employer groups that are dealing with just catastrophic rates of diabetes and prediabetes. So a lot of the public sector plans in our employer groups in our sample, like West Palm Beach School District and City of New Orleans, they were really drawn to this benefit because they see that 30-40% of their population has prediabetes. And so, I think we heard from the sales reps kind of what was the driver. We would love to do the workdays analysis, but that's held at the employer level. And, United doesn't want us to bother their customers. So, you can only go so far with a partner like this, but they have been, United has been an amazing partner for us for a decade in many research projects. And, at the same time, I think, there's certain things that they're just, they put boundaries on. So.
Great. Thank you very much.
You're welcome.